From Microbe TV, this is Immune, episode number 79, recorded on April 30th, 2024. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, welcome. It is an absolutely spectacularly gorgeous day here in Ithaca. I don't know about you guys, but it's beautiful here. Finally.
It was beautiful here yesterday. It was like
that too, yeah.
80-ish and sunny, but today it's cloudy and 20 Celsius. Not so nice, but it's fine. Also joining us from Madison, New Jersey, Brianne Barker.
Hello, it's great to be here. It is basically the same here as it is where Vincent is. It was absolutely gorgeous yesterday. And, you know, I'll take this. I still see some sun out there. Could be a lot worse.
Yeah, I don't mind the clouds. It was the warm I meant. Yesterday was sunnier, yes, but it is still very warm and beautiful today.
Yeah, I am in Chelsea. That's what my weather thing says, the Chelsea neighborhood of Manhattan. If you like what we do on these podcasts, please support us so we can do more of them. You can go to microbe.tv/contribute. Microbe TV is a nonprofit in the U.S., so your donations are U.S. federal tax deductible. Today, we're going to hear about the Fountain of Youth from Cindy.
Well, that's a good title. I was thinking about the titles. I had a bunch of ideas, but the Fountain of Youth is a good one. Yeah, so this paper that we're going to talk about today caught my eye because it got a lot of press, as do things that make you feel younger. They often do that. The title of the paper today is Depleting Myeloid-Biased Hematopoietic Stem Cells Rejuvenates Aged Immunity. And this was published in Nature only like 2 weeks ago. The first author is Jason Ross, and co-first author is Laura Myers. And this comes from Irving Weissman's lab, as well as Kim Hassenkrug. They are at Stanford and the Rocky Mountain Labs in Montana. So why this paper? Well, lots of people, including myself, are aging, and we'd all love to know how to be more youthful. And it seems like a far-fetched idea to rejuvenate the immune system, but, you know, that's what people do. I looked something up that I knew I had seen a story, and I just thought I would raise it just briefly at the beginning of this. So some of us think about being more youthful. Some people do stuff about it, you know, injections and exercise and whatever else they want. But have you heard about this guy who is spending almost $2 million a year to try and look 18 again? He's 45. It's insane. So
what does he spend the money on?
He has a whole medical suite in his house and a team of doctors that they call it Project Blueprint. He takes 111 pills a day, has a very strict vegan diet and all this other stuff. It's just amazing. He's 45 and he wants to look 18. I think
How does he look?
I don't know. 18? He looks puffy. He looks odd and unusual because as you age, there are certain things that normally happen that you look your age. You know, we'd all love to have fewer wrinkles, but I don't know. It seems a bit extreme, but nonetheless, you know, there are these people who are going to extraordinary lengths to try and be youthful. But the reality is, go ahead.
I was trying to remember the exact detail. Yeah. So I get the ProMed mail.
Yes.
Emails. And there was actually an email yesterday, a case within the past week. I'm looking at it now. It looks like the email came on over the weekend. Of people who were actually trying to look younger.
The vampire one.
Who were, yeah, so it's they were getting plasma transfers. It's known as a vampire facial. And apparently there were a whole bunch of unlicensed places and there was a bunch of HIV transmission.
Yes.
Because of plasma transfers from younger individuals. So I do no bad idea.
Bad idea. First of all, taking someone else's blood is not going to necessarily make you young. And we can talk about immune reactions and all of that. But the other thing is taking blood products from places that are not licensed is a very bad idea. For just that reason, because they're not licensed, they don't need to test these things. They don't test these things. And yeah, I read about that. That is terrifying. First of all, I don't want anybody to inject blood in my face. That's just weird. But yeah, so people think, getting stem cells, which we're going to come to, a point of this paper, would help you be more youthful.
My solution is it's all in your head. If you feel young, you will act young. Of course, that doesn't get around the immunosenescence, but
correct.
If people are worried about, and I mean, good genetics
yes,
takes care of that. And there you have no choice. If people are worried about wrinkles, they should have stayed out of the sun their whole life.
Yes.
Right?
Yes.
Because a lot of people are wrinkled because they were in the sun, and that's it. Or they smoke and their lips are all wrinkled around them, So
Yep.
there are things you don't need to spend $2 million.
That's right.
I stay young by podcasting.
But at least, all of this is, while wrinkles are a problem, I think really here we're thinking about the effects of immune responses or aging on immune responses.
Correct.
And so the idea that some older individuals have less robust immune responses, particularly adaptive responses to vaccines, and so are suffering more from certain infectious diseases, also maybe have increased inflammation. And so really this is less about wrinkles. I'm not sure if the treatment that they talk about here, they did not address wrinkle formation in the mice.
No, I just was raising that because it was a funny anecdote.
Oh no, of course.
Yes, we're talking about aging of the immune system, and you're absolutely right that I've read that it's around the 6th decade of life, which yay, fun. That's when things start to go south with your immune system. But if you remember, your immune system has a time clock from the beginning, because as we reach puberty, or even before that, the thymus starts to involute, and by puberty, it's involuted, right? So we can come back to that, because that's a very interesting point, because this idea of rejuvenating the immune system and influxing it with a bunch of new naive cells, B cells and T cells, could have its own issues. So that's an interesting thing.
So, Cindy,
yeah,
you're going to tell us exactly what happens to our immune systems as we age?
Yeah, so there are there's a lot of different things that happen. So there's an increase in various different biased hematopoietic stem cells that we're going to talk about. There's an increase in inflammation. Generally, it's called inflammaging, if you've heard about that. The innate immune cells tend to be more numerous and more inflammatory. Therefore, there's an increase in pro-inflammatory cytokines circulating in general in the more aged individuals. And there's, at each infection you have, you're going to generate memory cells, right? And you're going to have a less of a naive population of cells, because now you've got more memory cells, and there's only so much space in the body, right? So you need to keep some of these things around, and eventually you're going to lose some of these naive populations. And in fact, one thing that's been shown is in adults, when you have like chronic DNA virus infections, that over time, like for CMV, a vast majority of your T cells will be specific for CMV when you reach around 80 or so. And this is a sort of a memory, it's the idea of memory inflation. So more and more memory cells accumulate, they're biased towards the things you see more frequently, and you lose a lot of those naive cells that would be protecting you from new things you'd be exposed to.
And you could imagine that is both based on the accumulation of memory cells and maybe changes in the production of naive cells, particularly like with thymic involution that Cindy mentioned, where you might not be producing as many naive T cells. The amount of space for T cells is the same, and most of that space sort of gets filled up with memory T cells responding to those common microbes.
Right. And the overall outcome of all of those changes, including, increased numbers of memory cells, reduced numbers of naive C cells, reduced repertoire of naive B cells and T cells, inflammaging, is there's reduced wound healing. So most older individuals will know that it takes longer to heal. There's an increased susceptibility to cancer. We all know there's a lower response to vaccines. So once you reach a certain age, you're supposed to take a higher dose of a vaccine because it takes more to get a take of a vaccine to get the immune response. And there's generally just more inflammation. So more diseases associated with increased inflammation, including atherosclerosis, arthritis, neurodegeneration. So a lot of those are associated with the changes that are happening in the immune system. Now, if we come back to this particular paper, they would like to argue that one of the things that their lab has seen is that there is a bias towards the stem cells that are around as you age. So we need to talk a little bit about stem cells. So in your bone marrow, there are multipotent hematopoietic stem cells that in theory can generate every type of immune cell in the body, and including also red blood cells and platelets and megakaryocytes. So there are certain hematopoietic stem cells that have a tendency to make more red blood cells, platelets, monocytes, macrophages, and neutrophils. And these are called myeloid-based HSCs or hematopoietic stem cells. So they call them MyHSCs. I think it sounds a little weird if we keep saying MyHSCs. I'm going to try and say myeloid HSCs. So I just think it sounds weird. But then there's another set of hematopoietic stem cells. So this is interesting to start with, in that a hematopoietic stem cell we would think of as just one single multipotent stem cell type that can generate all the different lineages. And what they're saying is that even in the bone marrow, even in adults, there's a bias, and there's actually two types of hematopoietic stem cells. One that generates all the cell types I just talked about, and the other one leads to more of lymphocytic cell types like B cells and T cells. And they call those balanced HSCs.
So when I was reading this paper, this idea of the balanced HSCs that can make both myeloid and lymphoid cells versus the myeloid HSCs was completely new to me.
Yeah, me too.
And so I had, a million questions about exactly what was going on, some of which they addressed nicely here, some of which I still have questions, going in the end. But so I guess that was actually my question was this something that like everybody else knew that I just hadn't come across, or is this new to you as well?
I did not know it either. Maybe some of our listeners, if they're HSC aficionados, knew about this, but I did not. And you're right, Brienne. So they're saying that there's these two populations, and one basically makes all of the cells, and one really primarily makes all of the myeloid cells.
Yeah.
And they posit that over time, you accumulate more of these myeloid type hematopoietic stem cells. And they also posit that that's bad because then you have increased numbers of monocytes, macrophages, and neutrophils, and that will promote this more inflammatory environment in the aged individual. And further, they propose that if they could readjust the levels of these two types of hematopoietic stem cells, that they could rejuvenate the youthfulness of the immune system by producing more B cells and T cells, which would provide more naive cells, but also then limiting the number of myeloid cells that you're making to reduce the inflammation. That's their whole premise of this paper.
Yeah, and the one other piece that, again, I had to like sit and think about for a while is you mentioned that they were rebalancing these two different types. And when they're trying to rebalance, their specific idea is that they want to reduce the myeloid cells, sort of reduce the excess of them. Yes. And they're expecting that that's going to rebalance because I don't know if either it like makes more room for the other ones to grow or kind of lets the other ones come back and be in balance. And so, part of me had wondered, well, is it about taking away excess versus injecting some other ones in? And they're really focusing on the taking away the excess. Right. With the idea that that's somehow going to have a positive impact on the ones that had been reduced before.
Correct. And I guess the only way I thought about it was that these are very slowly turning over cells, right? So stem cells in general, if you know, they both self-repopulate, but then they can go through a division where one repopulate themselves and one generate the next level of differentiation. And I guess the idea would be if you eliminate enough of the stem cells that you don't want, you give more room for those low proliferating cells to turn over and make more of their downstream precursor cells. But again, that's sort of hand waving. So a lot of what they analyze in the paper is frequencies, as we often do with flow cytometry. So of course, you're going to see changes in frequencies. When you eliminate one population, the other population is going to, quote unquote, go up because there's just fewer of the one. And now if you look at the percentages, it looks like more. They did do a little bit about quantifying some numbers in some points, but I don't know exactly how this would all pan out if you tried to develop as a therapy, which apparently they're thinking about. Which I guess this guy who pays $2 million a year would
He'd be all on
be
board.
one of the ones that steps up to the plate and says, inject me. So, all right. So how do you end up reducing a particular lineage of cells within the immune system or in the body in general? Well, one of the ways we could do this is antibodies. So if you inject an antibody to a cell type, especially in the blood, the antibodies will bind to that cell, and that cell will be eliminated from several different mechanisms, including antibody-dependent cell cytotoxicity, or even sometimes complement-mediated lysis, or it could sort of agglutinate them and have them cleared by the liver. But this is actually a common technique that's used in mouse studies, where if we want to find out if T cells are important for something, we inject in some antibodies that recognize the CD3 molecule on the surface of all the T cells, and then those T cells are gone, and now we can do whatever we want to the mouse, like infect them and say, hey, where are those T cells important? And usually they're pretty important, but for certain nuanced things, you can eliminate certain subtypes of T cells or other cells or neutrophils or whatever. So you can kind of deplete the cell of interest. Now, it's usually done in the blood, and often the tissue cells will remain resistant to that because the antibodies circulate more in the blood and they will eventually diffuse out into the tissue, but it's less easy to deplete those cell populations than it is from the blood.
Yeah, and this isn't like unheard of in human settings. So you mentioned it's usually used experimentally in mice all the time to deplete different cell types. There are a couple of clinical examples where people have tried to deplete particular cell types, particularly sort of blood cell cancers, because of the issue with tissue versus blood and things like that. It's not used nearly as commonly, but it's not like totally unheard of.
Yeah, so Rituxan or rituximab is an antibody that's to the surface, either CD20 or CD19 on the surface of B cells. And most people, although it's not fantastic, you can live without your B cells. So if you get this injection, it will deplete your B cells, which if you have lymphoma, will deplete the lymphoma cells as well as the normal B cells. Depending on how aggressive the therapy is, you might still have B cells left over that could then repopulate, or you just deplete all your B cells and then you're just antibody deficient and you would need immunoglobulin therapy. But yes, people do that. Here they want to deplete these myeloid-hematopoietic stem cells, again with this idea that's saying, hey, if we get rid of them, then we'll have fewer myeloid cells around and less inflammation, and maybe our immune cells can be rejuvenated and more naive T cells and B cells. But the first problem is you have to figure out how to specifically recognize those cells. If you have Rituxan and B cells, it's pretty easy because B cells express CD19, no other cells express CD19. So if you add CD19, you're going to deplete B cells and no other cells, right? It gets trickier when you're talking about cells that are not so down the line and terminally differentiated. The further back you go, the more in common the surface markers are. So that's the first thing. They want to find markers that are enriched or much more highly expressed on these myeloid hematopoietic stem cells compared to other stem cells. And they want it to be something that is on the cell surface, because if you add antibodies to something inside, that's not going to deplete those cells. And the third thing is they want to look at things that are generally shared between human and mouse. so that there's more easy translatability, because as we know, not all the markers in mouse are expressed in human. So you might have this great therapy that works in mice and fantastic and absolutely not translatable. And then that's not very helpful.
It's very helpful if you're a mouse.
It's very helpful. You can be a very youthful mouse.
Yes.
Well, you can go back to when you put the T cells into the mice
Exactly.
over and over and over and over, and they lived forever, right? Okay, so they have to figure out they have to first define these myeloid hematopoietic stem cells. And so this lab has done this before, and apparently they are generally high expressors of CD150. So hematopoietic stem cells in general in mice are what we call lineage negative. That means they don't express CD3, which is for, that was weird. Did I just blurb out for a second? But they don't express CD, so lineage markers are CD3 to recognize T cells, Ly6C or G, which will recognize some myeloid cells and neutrophils, CD11B, which will be macrophages specifically, and B cell markers, like we've already talked about, and a marker called TER119, which is on red blood cells. So first thing is they're all negative for all of those. And then hematopoietic stem cells also express something called KIT, c-KIT, SCA1, and they're usually what we call FLAT3 ligand or FLT3 negative, CD34 negative, and CD150 positive. So they have a whole beautiful, extensive gating strategy that they show in the supplementary data if any of our listeners are interested in that. But what they argue is that they express CD150, but that the myeloid HSCs are CD150 high, and the balanced HSCs are CD150 low-ish medium. And if you look at the data, it is not a really robust difference. It's one of those ones where if you are not looking at flow cytometry all the time, you just see one blop and they put a line right straight through the middle and said, these are high and these are low.
Yeah, if you would have asked me about designing or thinking about a marker that would be a good choice to use for depletion, I would have looked at these histograms and said, not these markers.
Correct.
Because I never would have guessed that kind of shoulder difference would work for depletion. So I was already like, really? Just from these shoulders.
Yeah, I was a bit skeptical myself. They did go an extra step, and they tried to mine available transcriptional data sets to look at other markers. And so the way they did this was they took these hematopoietic stem cells from aged mice and young mice to see if there are differences, and then see if those are different between these two populations, with a CD150 high and 150 low. And anyway, they went to great lengths to try and identify some of these surface markers. And what they found were CD41, CD61, CD62P, Neo1.
Along with CD150.
And along with CD150.
Which to me was the most interesting one.
Yeah.
Only because it's the measles receptor.
Oh. I didn't realize
or a measles receptor. And so
Yeah,
I was sort of like, hey, I wonder if there's something up with measles with this. But that's as far as I got in my thinking.
Is that why measles eliminates that's memory cells.
Right, exactly. Right.
Well,
you're pretty much exactly where I am.
That's a vaccine strain which hits CD46.
Ah, okay.
So the vaccine strain receptor is different from the wild type virus. So
okay.
I don't know where CD46
So does wild
is.
type virus wipe out hematopoietic stem cells?
That was my question too.
Yeah. I don't know about that, but it wipes out memory, as you know. Yeah.
Yeah, but memory would be way downstream, right? Because then
exactly,
okay.
and would actually be a reason why CD150 would be a bad target here.
Yeah, so that's right. The wild type receptor is CD150. Yeah.
Okay.
So I mean, it destroys B and T memory cells. So
that I knew, but those are very terminally differentiated cells that shouldn't be expressing this CD150, right?
Right.
Because they argued that this marker was pretty
It's super specific.
for the HSCs and not for any of the other
Right.
downstream components that come out, like the lymphoid precursors or the myeloid precursors or any of the downstream differentiated immune cells. Interesting.
Yeah, that
Hmm.
I was just like, huh, that's a measles receptor as I was reading it.
Huh. Well, I did not know that. That's very interesting.
Slam. Slam.
Yeah, slam. Okay. So they go on to show that the hematopoietic stem cells from aged mice have increased expression of these markers. So that might be why they thought that they were more reasonable to test. So maybe there's not a huge difference between the balanced hematopoietic stem cells and the myeloid hematopoietic stem cells, but as you age, those markers seem to increase even more, and presumably more on the myeloid hematopoietic stem cells. Therefore, it opens the window a little bit more than you think as far as the ability to use that marker to tag those cells and deplete them. So there was just a general correlation with age for the three markers and CD150 that they chose. So the NEO1, CD41, and CD62P. But I think that the first note I made to myself, and we kind of already raised this, none of these markers are exclusive. And that's the best case scenario when you're doing a depletion strategy. So inevitably, we need to think about, and they didn't really, I don't think they addressed it that clearly in their discussions, is that you will deplete things that you're not intending to deplete. And we don't really know what the impact of that will be. So I think that they left that a little bit murky.
Yeah, I mean, there are all sorts of, at least in the sort of tumor immunology field, ideas about having bispecific antibodies or things that could let you target multiple of them at once.
Right.
So maybe some very long ways down the road, you could try to get around that issue. But you're right, they do not really come to an idea of what specifically else is getting depleted.
So first we had to find some potential markers, and then we had to see if they were actually on these myeloid hematopoietic stem cells. But then the big question is, can you actually use these to deplete the myeloid hematopoietic stem cells? So they went through and developed a strategy for each antibody, and it is very in-depth and I would just say pretty comprehensive as far as I'm concerned. For example, even going down significant rabbit holes to say, okay, if we've used CD150 to deplete the cells, and then we're trying to see whether they express CD150, maybe they just downregulated that marker. And we need to have multiple antibodies that recognize the same target to then be able to see whether we can see that target if it's been depleted or downregulated or whatever. And I think without going into great detail, I think they did a pretty good job of showing that if they used one of these antibodies to deplete, they lost the cells expressing that marker, not just that marker. And these other markers that they identified were also lost. So that would suggest that it's not just downregulation, but they are in fact losing that population of cells.
Yeah, all of that, I thought they did really nicely. From some other work I know of from this group, I know they've also been working on antibody depletion protocols with looking at ways to stop the anti-phagocytic signal. So they combine that work here as well to really kind of design a depletion strategy that's going to work as best as it possibly can, with the caveat that Cindy mentioned of it might hit other cells too.
Yeah. So you raise a really good point. So they did test 2 additional antibodies to include in their mixture. And one was CD47, which as you just described is the antiphagocytic receptor. So it will block if cells are supposed to be getting taken up by macrophages to be killed. If you block that CD47, it will the cells express CD47 to prevent their phagocytosis, and if you block that, they're more easily phagocytosed.
I think they, in some of their at least like reviews, they describe CD47 as a "don't eat me" signal.
Don't eat me signal. Exactly.
And so you're blocking the "don't eat me" signal so that the cell can get eaten.
Yep. And they also used c-KIT, or stem cell factor receptor. So stem cell factor is a cytokine that acts on hematopoietic stem cells. So most of them express c-KIT. Again, that's on all the different hematopoietic stem cell down to a couple levels down of the downstream cells that come from the hematopoietic stem cell as well. But they used this scheme, and they determined that their optimal depletion scheme was antibodies to CD150, CD47, and c-KIT. And that very cleanly, well, not cleanly, but it significantly depleted the myeloid-hematopoietic stem cells without having an effect on the next downstream, or the myeloid, what am I looking at? The balanced hematopoietic stem cells, or the common lymphocyte precursors, and other cells that come from that lineage. So it seemed like it was a good strategy. And they did individually develop those and show all the data in the supplement and in the main figures about, how it happens if you deplete with the NEO or with the other markers as well.
I was impressed.
It was a lot of work. It's a lot of figures in this paper. Okay, so if you deplete, but there are not 100% depleted, what is the transcriptional profile of those cells that are left? So does it change if you look at hematopoietic stem cells that are residual with that conditioning treatment? So they call it conditioning treatment. We use the immunodepletion, compared to the age mice that have not been depleted. So they do gene set enrichment analysis, and they show that the hematopoietic stem cells from the mice that got conditioned were enriched in gene signatures of quote unquote young HSCs and balanced HSCs. And they were depleted in signatures of old HSCs and myeloid HSCs. So altogether, that suggests that they have biased the myeloid hematopoietic stem cells grouping towards a more youthful version. And that in general, if you look at all of the HSCs, you're biasing towards the younger or the balanced looking ones as well.
So you're sort of getting a response that looks less like the 6th decade and more like, I don't know, the 3rd or 4th decade or something like that.
Yeah. I don't know how early, but yeah.
Yeah.
Oh, I do know, because they called young was, I have it written down. Young was 8 to 12 weeks. And then old was 16 to,
they talk about
I found
young adult
this.
as 3 to 6 versus aged 18 to 24.
Yes. Yeah, I've had it right there. 3 to 6 months versus 18 to 24 months. Okay. So one of the other things I wanted to mention at this point is that most of the analyses they do is 1 week after depletion. That's not very long. They did some other studies to show that it's more long lasting, but I think it's just a caveat that we should all think about is that they deplete, and then after 1 week, they examine the bone marrow or examine these cells transcriptionally and functionally and everything.
If that guy who pays $2 million for whatever was getting this treatment, he would have to perhaps be treated frequently. They haven't shown that he could go a long time between treatments.
Right, exactly.
Well, when you get these anti-B cell infusions, it's typically quite often, right? I think you do every 2 weeks for like a certain number of weeks, then you take a pause, and then you do it again. So it's part of, if you want to be young,
You have to keep on top of it. You have to go to the gym, or you have to go to the clinic regularly. Yeah. All right. So they definitely saw these changes in the hematopoietic stem cells. There's no doubt about that. But so what? Does that have anything to do with the progenitor potential or the function of the immune system or anything? So the rest of the paper really kind of starts to address that part. So the next question they ask is, will changing this ratio, which they just showed that they could do, change the progenitor potential? So that means, is there going to be more T cells and B cells? Do you have reduced numbers of myeloid cells?
Mm.
So to do this, they transplanted purified HSCs from mice that had been treated or not into recipient mice, and then evaluated the generation of myeloid versus lymphoid lineages. So they take advantage of the commonly used method in immune studies is CD45.1 and CD45.2. It's basically just two different versions of the same molecule that are expressed in different strains of mice. There's specific antibodies that recognize them. So what you can do is if you park cells from mouse A into mouse B, you can see A cells and B cells in the same mouse, and you can distinguish them. And then you can ask, so if you park in hematopoietic stem cells from A into a B mouse, you can see whether the B cells came from A or the myeloid, macrophages came from A or not.
And you can sort of see if the A cells and B cells are responding similarly or if they respond differently.
Right. Yeah, you can actually do functional analyses and look at those two cells by isolating them. So I think it's cool when they do hematopoietic stem cell stuff. They put 100 cells. 100 cells. That's it. You have to count them very accurately, dilute them, 100 hematopoietic stem cells from a CD45.2 mouse that was 11 months old that either had gotten this conditioning treatment or not. And they put them into the 2-month-old mice that were CD45.1 and ask, what kinds of cells do you get out? And they found that the mice that received the hematopoietic stem cells from the conditioned donors, so depleted of the myeloid hematopoietic stem cells, had decreased myeloid to lymphoid cell ratio. What does that mean? That means that those cells, you harvest the bone marrow, harvest those hematopoietic stem cells, and you take 100 of them and you put them into a mouse, there's more, this is the interpretation, there's more of the balanced hematopoietic stem cells versus the myeloid hematopoietic stem cells, therefore leading to the generation of more lymphoid cells and fewer myeloid cells.
So this, and the control is untreated mice, right?
Correct.
Yes.
And in those mice, you don't get a correction of the imbalance,
Right.
Mm-hmm.
Okay.
Yeah. So that's great. You can take those cells and you can put them into another mouse and you can look at them differentiate. And I thought that was really important. But then they also wanted to actually deplete in the same mouse and then look at the effect on the bone marrow. Again, this is a limited time span. It was 1 week after treatment. But what they did was do the depletion or not, and then look at the repertoire of cells that come out of the bone marrow in those mice. And what they saw was there was an increased frequency of the common lymphoid precursors, suggesting that the balanced myeloid hematopoietic stem cells generated more of those common lymphoid precursors. So there's a bias towards generating T cells and B cells in the bone marrow from the mice that received that conditioning. So again, altogether, this says that, one, they have markers that generally sort of specifically recognize the population of interest. They can deplete them successfully. And when they deplete them, there is actually what they expected to see, which is there's more lymphoid cells and fewer myeloid cells. So that all fits so far with their model. But again, so what? Does that do anything to the immune response? Because we started out by talking about the thymus is involuted in these aged mice. So you're not going to have education of T cells anymore. You will have an influx of new B cells, and then they could be responding to new antigens. But does it actually matter or not if you challenge the mice? So in this case, they took young mice or aged mice that have been conditioned. Now, I am going to raise one issue here, and that is you had to pay really close attention to which depleting strategy they were using, because they came up with what they thought was the optimal depleting strategy in one figure, and then they use a different depleting strategy in a different figure. And in this one, they use a neo, not the CD150, or whatever that's worth, but they use the neo-conditioned mice. But then they test it 1 week and 8 weeks. And what they showed was the aged mice that received the conditioning treatment had a decrease in the myeloid hematopoietic stem cells as we expected, but no significant effect on the frequencies of mature B cells or myeloid cells, which is interesting because that wasn't fitting exactly with what they expected. But 8 weeks after treatment, the frequency of myeloid cells to balanced hematopoietic cells was still significantly reduced. And if they looked at the young mice compared to the untreated old mice, there was definitely a reduction in the frequency of common lymphoid precursors and an increase in the myeloid precursors. That the conditioning flipped that. So what does that mean? Let me step back because that was a lot of information. So basically, when they get older, they have more of the myeloid precursors and a decrease in the common lymphoid precursors. If they do the conditioning, the older mice that got the conditioning, it flips back to what it looked like when they were young. So there is this change in the precursor frequencies in the bone marrow. But then they looked at the numbers of lymphocytes. So they also evaluated this after 8 weeks. And they looked at the generation of new T cells and B cells at 8 weeks, because apparently the peak of hematopoietic stem of generating new T cells and B cells from hematopoietic stem cells is about 7 to 11 weeks from prior studies. So that's why they looked at 8 weeks. And they say, although we did not observe any difference in the thymus weight, treated mice contained all of the thymic progenitor subsets associated with thymic function. So that's interesting. I didn't not entirely know what to make of that.
Yeah, I was thinking about that as well. And my question was sort of in terms of how old the mice were at the beginning, because I'm not sure that you would expect the thymus weight to change all that much because you're too late in the process.
Right. So I think in mice it involutes quite young.
It really does.
By 3 weeks or so at weaning?
Or
a little bit after that.
A little bit after much. That. And I sometimes get a little picky about people who imagine our imagination of involution as like thymus is 100% and then to none.
Gone, right.
So I think it's a little more gradual than we sometimes make it out to be.
Yeah.
But I do know that when I tried to do experiments where I get thymocytes, I have to get very young mice, and I know how hard it gets very quickly as those mice get older. And so my thought was, well, is it just because the mice were already too old for you to really see that change?
It could be. Yeah. But I guess the good news is that after 8 weeks, the mice that had the conditioning had increases in their frequency and numbers of naive T cells, both CD4 and CD8, compared to the age-matched controls. So according to that, they have more of them. Now, given this whole discussion around thymic involution, are they educated properly is a whole nother issue, right? The job of the thymic education is to get rid of the cells that are autoreactive and make sure that you get out the cells that are useful and functional and have a good T cell receptor. I don't know exactly what we're getting out of this at this point and how they're educated, whether they're extra, generally extra thymic T cells at this point, or what exactly is going on that they're generating. But it's definitely not going to be the same T cells that we are normally accustomed to.
Yeah, maybe it might be interesting to take mice that have had this conditioning and let them age out further and see if they start to have more kind of autoimmune issues.
Right. They did not do that. They only went 8 weeks. So I don't know. They did go into excruciating detail of all the different subsets of T cells and memory T cells and all of these markers, effector memory, central memory, and so forth. And basically there was a slight increase of central memory cells, but the absolute numbers were not really affected. So it was pretty inconclusive, I would say.
Yeah.
They did see it also increase in the frequency and absolute numbers of mature circulating B cells. So IgM, naive mature circulating B cells. So I guess we conclude that they did make more T cells and B cells, but exactly what the function of those cells are was not clear from those particular studies. So how do you test whether there is any function? We know they looked at one thing, which is exhaustion markers. So one of the other things that happens as the immune system ages is the cells get tired, basically, and they upregulate markers of what are traditionally called exhaustion. And people can argue back and forth of whether these are true exhaustion markers, what exhaustion means, because a lot of these markers are also activation markers. So there are exhausted cells, but exactly what markers they have and whether these are the right ones, I think could be a point of dispute. But what they see is that in aged mice, the CD4 T cells with the exhausted phenotype increase, and antibody conditioning flips that back to looking more youthful again. So again, we've got T cells. Whether those T cells are the ones that got generated from now the hematopoietic stem cells that are more lymphoid biased, and then they're generating new T cells, and whether those are these T cells or not, I'm not entirely sure. So there might be very complex things going on here, because when we think about the aging of the immune system, there's the aging of the cells themselves, and there's an aging of the microenvironment, and then the microenvironment can influence those cells. So exactly what's going on here, I'm not 100% sure.
I will get more into this because there's an experiment that they didn't do that I was kind of like,
oh,
at this point I was like, I want to know the answer to this experiment. And at this point I was just like, where's my experiment? That's not it. Where's my experiment? That's not it. As I was reading. But my short answer that sort of is really getting at your point as well is that there's also a situation where an aged cell might be making different products that could influence other cells.
Correct.
And so it's possible that these mice have better T cells because they're the young T cells, or it's possible because these mice have better T cells because they're not getting some sort of bad signal from other aged cells.
Yep, like the myeloid cells that we know are more inflammatory that can affect the T cells.
Yeah,
agreed. What
is that an experiment you could do?
So,
yeah.
Maybe. I think that there are some ways that you could do some tricky transfers here to look at that, where you actually tried to look at, say, if just, if you're transferring, then can you actually mark the T cells that are CD45.1 versus CD45.2 and see if there are changes in exhaustion between the two groups? Are the new T cells not exhausted, but the old ones are, kind of thing. My experiment that I was so curious about throughout this whole paper, and it's entirely possible that there are other papers that address it, but it wasn't addressed here at all.
Yeah,
were about the myeloid cells that come from these two different types of HSCs.
Yep.
And so I wanted to know, are the myeloid cells that are generated different myeloid cells or not? And so I could imagine if the myeloid biased HSCs make like messed up myeloid cells, then those messed up myeloid cells could be producing products that are causing many of these aging effects. And so the issue here is not that the T cells actually themselves are changed. The issue is whether or not they're around the bad myeloid cells. And so I really wanted to know if these two hematopoietic stem cell types made different myeloid cells, perhaps based on gene profiling, and if the conditioning changed those myeloid cells that are made, those myeloid products, I guess is my best word I can come up with right now. And they don't really address what myeloid cells come out of these two groups at all.
I bet there would be a way to fate mark once you become, I know maybe there isn't, a myeloid versus a common lymphoid, or the the
The
balanced.
balanced.
I don't know if there's enough gene differences that would be specific at that point, but you might be able to mark one step down from that, where you would at least be able to see the T cells that came, or how would you do that? Yeah,
Well, but they
maybe
do-
there's a marker that's turned on the next stage down that would be able to mark them.
Well, in theory, you could
Yeah.
you could do the transfer,
Transfer. Yeah.
and then you could take the CD45.1s that have differentiated into myeloid cells out.
Oh.
So you take out, so you move the stem cells, let the cells differentiate in the new mice, and then take out the differentiated cells and say, do gene expression profiling and say, are these myeloid cells the same kind of myeloid cells?
Got it. Yeah. So you'd have your myeloid HSCs and your balanced HSCs on CD45.1 or 45.2 and park them into another mouse.
Transfer them.
And then pull all the monocytes out or myeloid cells out and see if they're different.
Exactly.
Ha. Interesting.
I was like waiting for that and was so curious about that the whole way through the paper, and was getting sadder and sadder as I read and saw that it wasn't there.
Yeah. Well, even
there's so much work. I am not criticizing the authors. I just thought that was good. I was really excited to know the answer.
Yeah.
Well, I think this paper will stimulate people doing things like that.
Yeah.
There's a lot more to do, as you can tell, but I think you can't do it all in one paper.
Of course not.
You can't do it all in one paper. But if we step back, basically what they showed over and over was they're like, this is associated with age, and here's the young, here's the old, it goes up, and if we condition the mice, it goes back down. So that's T cell exhaustion markers, B cell marker, aged B cell markers, inflammatory cytokines. It was really quite clean how they presented it. This goes up with age, and guess what? If you condition them, it goes back down. So I thought that was pretty convincing for what they are arguing are age-associated changes. Really, the conditioning did, quote unquote, youthify or whatever, make the mouse more youthful from those types of things. But again, like you step back and you say, okay, so what? The most important thing is, does it protect us from infection?
Mm-hmm.
So you guys need to help me with this one. So they used the friend virus, which is not one that I can say I was all that familiar with. I got that it was a retrovirus. They use a live attenuated strain as their vaccine strain. And apparently, if they use enough, it's pretty pathogenic. But is there more we should know about friend virus?
Isn't friend virus a type of MLV?
Yeah, it causes leukemia in mice, yeah.
Okay.
Mm-hmm. Discovered by Charlotte Friend at Mount Sinai.
It's interesting if it's a lymphoma-inducing virus, because now they're looking at T cells and B cells. And okay, that's interesting. We should think about that. Regardless, they vaccinate their mice. IV. Do you normally do an IV vaccination? I was very surprised by that too.
No.
Right.
That wouldn't be normal at all, right?
It's the second paper in 2 weeks that I've talked about. At our journal club that we had here, there was a paper that was looking at intravenous BCG as for immune training. And it was odd, I thought.
That one I felt like I feel like there is an intravenous BCG literature.
Yeah.
But I've never heard of intravenous friend virus.
I don't know. Anyway, we'll just let it go. They did IV vaccination with a live attenuated virus. After about 8 weeks, then they did the conditioning. So the idea is if you've got a young mouse and you infect them, they get sick, whatever, and they're okay. You have an old mouse and you infect them with friend virus, apparently it's much worse. And they're saying, okay, if we condition these mice, are they more youthful? Do they defend against this virus more? So they have aged mice. They give them the vaccine. They condition them with these the Neo-1 mixture, and then they collect the spleens after 10 to 14 days, which they say is the peak of the CD8 T cell response. And the aged mice that got the depletion had increased virus-specific T cell response. Mm-hmm. And they also had a better antiviral response. with challenge. So with the challenge with the friend virus, they measured two things: splenomegaly and infectious centers per spleen. That was also something I wasn't entirely familiar with, but apparently that is sort of a count of virus load.
Right.
And I was surprised. So if you infect a young mouse, they get some splenomegaly, a little bit, and they get 7,000 infectious centers per spleen. But if you challenge an aged mouse, they will get gigantic spleens and over 4 million infectious centers. So when they conditioned the mice, those aged mice that got conditioned were significantly better off and more youthful-like. They had less splenomegaly and fewer infectious centers that were closer to the young mice compared to the non-conditioned old mice. So this would suggest that their depletion actually worked. Their strategy worked, at least for this one type of infection. They were able to generate a better immune response if they depleted these myeloid hematopoietic stem cells from the aged mice.
It's an interesting choice of virus.
Why not flu or something we know infects
Well,
older people
it's a mouse
worse?
virus. It's a real bona fide mouse virus.
Fair enough.
So there is probably a good argument for doing that.
Yeah, the other argument I wonder about is that it's pretty dramatic between the sort of younger mouse and the aged mouse. So they have a big
it is huge
range
difference.
where they could try to get a decrease. And so maybe with some other viruses, the difference is more subtle. And so it's harder to show that you got a decrease.
So you want an age-dependent difference.
You want a super age-dependent difference.
And if you do, so there's no age-dependent COVID mouse, really. So that's probably why they did this. I think it's true. And then you can move on to others as well. But, I don't know any other human virus that would have such an age dependency like this in mice. But there might be one. So that's probably why they did it, yeah.
I did have one quibble with them about their data presentation is they left out a control that I thought they should have had. So when they show the conditioned mice, they show their uninfected spleen, right? And then they show when they are vaccinated and infected, but they don't show what infected alone has. And they do show that for the young mice and the aged mice. And that's where they saw this big increase. So the young mice have a normal size spleen. You infect them, they get a big spleen. If you vaccinate them before infection, the spleen doesn't get big anymore. If you look at the aged mice, they have a certain size spleen. You infect them, it gets gigantic. And if you vaccinate them, it goes down a little bit, but not that much. And when they show the conditioned mice, they just show uninfected and vaccinated infected. And they're basically the same. But they don't show you how much it goes up with just the infection alone. And I wonder whether they have a less of an increase in general, which it just, this is interesting that they just decided not to show that.
Yeah, that is interesting.
But, all right. So in general, this conditioning treatment, you can identify markers on the surface. You can use antibodies to deplete these cells. When you deplete these cells, you change the ratio of myeloid hematopoietic stem cells and balanced stem cells. That leads to changes in the frequencies of myeloid cells and B cells and T cells. That leads to changes in function, and that leads to changes in differential susceptibility to infection. So all of that's really great, but so who cares if there is nothing translatable to human? They do go the extra step.
Mice care.
Right. They do go to the extra step, and they show that the human homologs of the marker genes that they identified and used in this study are actually increased in human hematopoietic stem cell transcriptomes. They looked at a bunch of different ones, and they validated that with antibodies looking at human hematopoietic stem cells. And in this case, the markers are a little different for human hematopoietic stem cells than mouse. So if you're an aficionado, they're Lin negative, CD34 positive, CD38 negative, CD90 positive, CD40RA negative. And then they looked at their different markers, including the CD150, and they saw that they were up in aged hematopoietic stem cells. So all three markers are They call them potential therapeutic targets for myeloid-hematopoietic stem cells depletion in vivo that are present on a subset of human hematopoietic stem cells. Have they validated that those are actually myeloid biased stem cells in human? I'm not sure if they've shown that in other studies. But it's intriguing to think that maybe one could think as a new type of therapy to mess with the frequencies of different hematopoietic stem cells to bias the types of immune cells coming out the end that then might influence the function of the immune system in general to infections and so forth. The news and views that came with it had one thing that I thought was pretty important, and it says, however, antibody conditioning could, in fact, exacerbate the emergence of clonal hematopoiesis by narrowing the total repertoire of hematopoietic stem cells. Depletion of myeloid-based hematopoietic stem cells could force a compensatory expansion of the remaining HSC pool and confer a selective pressure on residual HSCs harboring advantageous mutations. Ultimately, this could accelerate clonal dominance and could progression to cancer. So everything good comes with a flip side, and it's possible that if you bias towards these balanced hematopoietic stem cells that have a higher turnover proliferation rate and generate T cells and B cells, you could lead to different cancers. Or if you deplete the myeloid hematopoietic stem cells, the ones that survive that conditioning might be biased towards potentially generating some cancer type phenotypes.
Yeah, it's sort of like you're giving even more room for one that has an advantage.
Correct.
To outgrow.
Yeah. So I think more work needs to be done, but I thought that this really was intriguing in the idea that I had not seen someone think about, should we mess with the repertoire of hematopoietic stem cells and potentially change the functional outcome of the immune system, and whether that was related to aging.
I didn't even know there was a repertoire.
I didn't either. I thought they were all one, and then they make myeloid precursors and common lymphoid precursors. Then it went from there. So
exactly.
I've revised how I think about hematopoietic stem cells at this point.
Yeah, it's interesting that there are a number of companies out there that are working on aging, right? Including Altos by Jeff Bezos, right? He has attracted a lot of high-profile scientists there. And I don't know. I was just looking at the funding. They didn't support any of this. I wonder if any of them have looked at this, because we don't know what they're doing with those companies.
Nope.
But some of the approaches were sirtuins, right, which make yeast live longer, and those have been explored in mammalian models too. But I think this is a really interesting twist, as you say. I think it's cool, and it's not ready yet, obviously, but it's very provocative.
It is. I mean, it's fascinating, and there's a lot of great immunology you can learn from thinking about this.
For sure. But don't go out and do this, folks.
Don't go deplete your myeloid hematopoietic stem cells, please. And don't go get vampire facials.
No, please, especially not like unlicensed.
Right.
Yeah.
Well, thank you, Cindy. That was cool. All right, that's Immune 79. You can find the show notes at microbe.tv/immune. If you have any questions or comments, immune@microbe.tv. And if you enjoy these kinds of programs where we give you the science straight from the journal, as you see, we'd love your support. Microbe.tv/contribute. Cindy Leifer's at Cornell University. Cindy Leifer on Twitter. Thanks, Cindy. Brianne Barker's at Drew University. BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
And I am Vincent Racaniello. You can find me at microbe.tv. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.